Transgenesis can be defined as modification of the genetic of an organism through recombinant the genetically modified animal to study gene function and regulation of genome mapping. We use test knock-in gene or knock-out gene. We get the gene inside the organism and we see what will be, and or we get the gene outside the organism and mice or rabbit and see what will be this organism. In cancer research, the gene can be modified to jump around willy-nilly around cells by putting them into a batch. Identify which gene or combination of gene lead to the cancer. Jump gene from jellyfish by putting them into a batch of human or mouse cells and watching to see which one become cancerous. Development of the model animal. Model is something that resembles something else. Cystic fibrosis, mouse, cystic fibrosis, multiple sclerosis have been made using animals. Medical advances over the last centuries have been made using animals. Biomedical research model for disease to learn about and test and treatment before attempting to diagnose. Benefit of the transgenic phytase enzyme in poultry. Salmon, improve gain efficiency and protein production. Super salmon, antifreeze protein and flounder and chinook salmon. Human therapeutic protein, anti-prothrombin, atropine, antiferrin, monoclonal, polyclonal antibodies, serum albumin. General strategies: embryonic stem cells, nuclear transfer, DNA vaccination, ex vivo transgenesis. The embryonic stem cell, the outer cell, and this is the inner cell. 5-6 blastocysts isolate the inner cells. The promise of stem cell research, drug development, toxicity test, experimental tissue and cell for therapy, cultured pluripotent stem cell. You can get bone marrow, nerve cells are diseases. Sheep, black-faced sheep, remove the nucleus, donor egg, and fuse the cell and get egg fused with the cell. We get the embryo, implant the embryo in a foster mother, and we get the black-faced sheep with white-faced lap. Nuclear transfer, lamb 6 LLC, dolly, donor, adult mammary gland cell, fine dorset U, recipient, inoculated oocyte, Scottish blackface U, the mammary epithelium, we get the cell, induce, go phase, inhibit the growth, fuse, activate, we get the cloned animal, unique mice. In 1981, first transgenic mouse insertion of HGH into a mouth, production of monoclonal antibodies, anti-inflammatory agents. This has been the most common method. Donor animal, donor cells culture, clone, nuclear transfer. This is the donor animal, donor cell, we take donor cells, and mature oocyte, inoculation. After that, nuclear transfer, cell fusion, cloned embryo developed. The human gene responsible for producing a desired protein is isolated and wrapped in the laboratory. An animal is given hormonal treatment to produce large number of embryos, and the embryos are collated from the oviduct. The human gene is inserted into the fertilized egg via microinjection. The transgenic embryo is placed in surrogate host, which give birth to the transgenic animal. The offspring is tested for new gene. Gene transfer is the production of transgenic dairy farm. Transgenic master control bacterial genes, bacteria gene for Staphylococcus aureus producing lysostaphin, a protein that kills bacteria Staphylococcus. Transgenic goat produce human lysozyme in their milk. Lysozyme is a protein found in the tears gene for an antibacterial enzyme found in human breast milk. Lysozyme inhibits the growth of bacteria by destroying the bacterial cell wall. Treatment of infant diarrhea. Transgenic goat milk contains a protein that could be extracted. The genes that produce antithrombin build a synthetic gene and make many copies. The synthetic gene is then attached with a casein to the gene for ensemble is promoter gene already injected into the newly fertilized egg. During the first few critical cell division, the gene may become attached to the goat DNA. Then if successful, the new gene is now transgenic, which will become incorporated into all cell during subsequent division. Then the embryo is then transferred to a surrogate mother. The offspring is the target. Cloning through the age in 1952, cloning by a nuclear transfer in frog in 1990, first mammal clone. 1995, first clone by culture mammalian cells. 19 cloning by adult cell dolly the sheep. First clone by transgenic alpha cells poly. 2000 cloning of cattle pig mice goat. First reported cloned human embryo